Okay, so week 8. So last week you learnt a little bit about, well, I say a little bit, a lot about cancer and a bit about blood components. So that's what we're going to go through this week. Hopefully it all kind of made sense, but if not, it's all good. We're going to be going through it. Yeah, so we're just going to start off with talking about cancer. So what is cancer? So cancer is basically abnormal or uncontrolled division of cells, and they can invade and destroy surrounding tissue. This is actually slightly incorrect. Sometimes they can be benign, sometimes they can invade and destroy surrounding tissue. Um, Normal cells become cancer cells because of genetic errors. So usually 4 to 6 errors are needed before the cell becomes cancerous, and these mutations generally affect how the DNA is transcribed or how it goes through replication. So it's not as controlled as it usually would be. So it just it's like, yeet, I'm just gonna make 20,000 copies of myself, and it's gonna get more and more mutations each time. We love that. So that's how cancer gets worse and worse. So there are two ways how these mutations can occur. So you have acquired mutations, so that can be environmental factors like radiation or viruses, bacteria, or asbestos and chemicals and things like that. And some can be inherited, which affects 10% of cancers. I think they like you to remember that stat. I feel like it was kind of drilled into my head a little bit that it's 10% of cancers are inherited. I don't know if they really examine it, but fun fact to remember, 10%. So the process of normal cells transforming into cancer cells is called transformation. Kind of makes sense. And progression of cancer can depend on the type of cell it originated from. So some grow slower and some grow faster. So breast cancer, I think grows reasonably quickly. I want to say. I think quickly. I think there's a lot of vasculature and lymph around there as well. Don't take my word for that though. I will Google it and maybe get back to you on that. So now we're going to go through some features of cancer cells, and there are quite a few of them. But a lot of them kind of make sense if you think about it. So it's basically all these reasons why cancer cells replicate quickly. So the first thing is an enlarged nucleus. So that kind of makes sense because they have a lot of mitosis, and the mitosis isn't always controlled, and you end up with a lot of extra DNA that shouldn't be there. Like in this karyotype here, you can see that instead of having 2 of each one, they have 3 of each one or 5 of each one. So obviously this is going to enlarge the nucleus because there's more contents. And then you have accumulated genetic errors and chromosomal aberrations. Same thing. You have lots of replication. Lots of errors happen with each replication, and they just increase as you have further generations of cancer cells. They also tend to be quite immature because they're replicating at such a fast pace. They don't really have time to specialize, how in G0 and stuff, they kind of go off and specialize and stuff. They don't really have time to do that because they're just time to replicate again. So we just got a question: what are aberrations? I mean, aberrations are just basically, it's the same thing as errors. I think it's just a fancy word for errors. Aberrations are basically just, yeah, pretty much chromosomal. It'll be chromosomal abnormalities, I think is basically the same way of saying it. Yeah, you also don't have any contact inhibition. So if you think about it, usually, yeah, because this kind of I was like, okay, I get it, but I don't understand why. So a lot of the time when you have things growing, they all grow on a basement membrane, right? It's on your skin, you have a basement membrane, and then you have all the different layers. And usually, everything in your body kind of has a basement layer, and then everything grows. But for cancer, they don't stop when they come into contact with each other. So it would grow on the basement membrane, and same thing, these two are kind of very similar, no contact inhibition and no anchorage dependence. So you would have instead of it growing to fill a space and then stopping, it grows to fill the space and then keeps growing and it keeps growing. And because it doesn't have any anchorage dependence, it can detach and flow around the lymph or the bloodstream, and that's how metastasis occurs. Because I mean, usually you wouldn't get a bit of skin just floating through your bloodstream to another random place. It doesn't really work like that, because it would die. But this cancer cells can survive. Don't we love that? Yeah, so they also don't need as much requirement for external growth factors. So usually in normal cells, when you have mitosis mitosis needs to be like enabled by external growth factors that kind of tell the cell, yes, it's okay, you can continue, you're at the correct size, or like there are no mutations, so yes, you can continue and divide. But cancer cells don't do that. They can produce their own growth factors, and a lot of the time, even when they, I think Jess probably touched on it a little bit in like immuno, in this next one. Wait, sorry. I missed change the cytoskeleton. Changes the cytoskeleton to allow for cell movement. So like, the cytoskeleton helps the cell move along and stuff like that. But resistant to inhibitory growth signals. So like I think Jess mentioned in immuno that there is like a particular receptor that usually like if like a gene doesn't possess it, then immune cells would come along and like destroy that cell. But cancer cells are quite smart and they show this receptor, so the immune cells come along and it's like, oh, that's okay, you've got that. Okay, we'll go along and not attack you. And also, like, if the body's just like, oh, you should stop growing because you're growing out of control, it'll show these receptors and it'll be like, oh, okay, you're OG, and just, go away. But they are resistant to that, so that is why they have uncontrolled division. Another thing is they have active telomerase. So, like, you'll hear Craig Hassard go on and on and on and on about telomeres. It's a bit of a meme. But it's because usually, like, in the human body, telomeres cap chromosomes with like a little bit at the end, and they're telomeres. So they cap the ends of the chromosomes, and usually they just kind of degrade as you age, which is what causes some, things about aging. But telomerase is active in cancer cells and just keeps replenishing these telomeres, which usually get like they slowly get shorter and shorter with cell division. But cancer cells can just replenish their telomeres, so they never apoptose and die because they always have telomeres. Yes, and then escaping immune surveillance is like what we just talked about. And here's just a little table of just kind of the main ones that are important, I guess, and kind of a comparison. Now, what was I going to talk about here? I think, okay, yeah, this is just talking about like how mutation kind of accelerates cancer development. So you have your normal cells, and then you have this one mutant cell, and you're like, okay, one mutant cell's not too bad. And then that mutant cell creates more mutant cells, and you're like, oh, that's not good. And those mutant cells create even more mutant cells. And because they're proliferating and getting even more mutations, then those cancer cells become even more invasive and even more like cancerous, I guess. So as the cancer continues, the invasiveness of the cancer can increase. Development of cancer. So these three types of genes are quite important. So you have oncogenes, tumor suppressor genes, and mismatch repair genes. And I remember being like always getting them confused. I was like, what do you mean? Tumor suppressor genes, oncogenes? I don't know what these words are, but you keep saying them. But the simplest explanation is oncogenes promote cancer, tumor suppressor genes prevent cancer. So it kind of makes sense if tumor suppressor genes prevent cancer because they're tumor suppressors, and oncogenes promote cancer. So onco is like oncology, so it's cancer. So if they're oncogenes, cancer genes, they will promote cancer, if that makes sense. So the thing is usually, so you can have two types of mutations. You can have loss of function mutations or gain of function mutations. So if you think about it, if you have a loss of function of a tumor suppressor gene, genes that act to prevent cancer, then you will have a higher likelihood of developing cancer. If you have a gain of function of an oncogene, then you are also more likely to develop cancer. So it's basically like what mutations affect what genes and what effect does that have. And of course, you also have your mismatch repair genes, and they'll basically repair mutated DNA, which is obviously important because otherwise, if you have mutated DNA, then it increases likelihood of like uncontrolled cell division. So oncogenes, you have oncogenes and proto-oncogenes. The way I liked to think about it is that oncogenes is also on. So like if you think about like driving a car, if you have an oncogene, your foot is on the pedal, so it's accelerating. whereas your proto-oncogenes are like your brakes, so it prevents it. Yeah, so proto-oncogenes are completely normal, so they're usually in your body, and they promote cell growth and cell division, so that's nice. That's good. But then oncogenes are when proto-oncogenes mutate. And that is what always got me, because it sounds like proto-oncogenes come from oncogenes, but actually oncogenes mutate from proto-oncogenes. But that kind of also makes sense now that if you think about it, because proto is kind of like before, if that makes sense. Proto-oncogenes become oncogenes. It's like a pre-oncogene, if you want to think about it that way. Yeah, so oncogenes are mutated forms of proto-oncogenes, and they promote inappropriate cell division, predisposing to cancer. So you have one-hit hypothesis and gain-of-function mutations. So a one-hit hypothesis versus two-hit hypothesis is basically the idea that, like, with your oncogenes, so if a one-hit hypothesis means if you have one mutation, that will already like let cancer go crazy. Two-hit hypothesis means you need two mutations, so you need both alleles of the same gene to be mutated in order for an effect to occur. So oncogenes are a one-hit hypothesis. So if you have like your two alleles of proto-oncogene, and one of them becomes an oncogene, that's already like you could get cancer. And it's a gain-of-function mutation. So proto-oncogenes are usually quite normal. They just promote cell growth. And as soon as they become an oncogene, they gain more function than they usually would, and they over-promote cell growth and cell division, if that makes sense. Yeah, so gain of function, so it does more than it should. Yeah, so this is just an example of an oncogene. So you have a normal cell, you have a single mutation event, does this, creates an oncogene, and then that can simulate cell proliferation. Now you have tumor suppressor genes. Now your tumor suppressor genes are quite actually normal. You're meant to have tumor suppressor genes, and they're present in all cells. But the problems occur when you have loss of function mutations of your tumor suppressor genes. So your tumor suppressor genes are an umbrella term for like all the genes that control the cell cycle, and they generally negatively control the cell cycle. So when it basically just it produces inhibitory proteins to thus prevent cell cycle from occurring. It's like when we talked about cell cycle way back in like week 2 or whatever, and you have all your like CDK and like CD like blah blah blah, all those annoying things that you have to remember. Those things are probably produced by tumor suppressor genes. So like if you didn't have those being produced, then the cell cycle would just go out of control. So because of that, it's a loss of function mutation, because it normally is producing things, but now it's not. So it's like reducing what it usually does. And this is caused by two-hit hypothesis. So both of the alleles must be mutated, which is good because it means, it's harder for you to get, the loss of function mutation. Loss of one allele does affect it slightly. It slightly increases cell cycle progression, but like most of the time it's like okay, like it doesn't really affect that much. But as soon as you lose two alleles, there's complete inactivation, predisposes to cancer, all that shit. It's very bad. And also, if you are born with already only one functioning allele, so if you inherited like a mutation from your parents, then obviously that increases your risk of having cancer, because it's easier for you to get that one mutation instead of two, and then cancer. Mismatch repair genes. I mean, it kind of is in the name, mismatch repair. They repair mismatched DNA. And often when you have cancer, the MMR genes are lost or defective. And these are two-hit hypothesis and loss of function mutations, because obviously if you had the function of repairing mismatches and then you don't have the function of repairing mismatches, you have lost function. Okay, here's a quick question. What is the two-hit hypothesis? Okay, we've got a quick question. Are those the genes that do the mismatch repair mechanism? Is that just like generally for all DNA, like transcription and stuff? Because I would say so, yes. Because if they're, I wonder if I can go back without showing the next question by accident. So, I would say so, yeah, because usually they're like functional and normal. So in the normal cell cycle, yeah. So like if you don't have so yes, in the cell cycle notes, it does happen after DNA replication. But like if you have replicated your DNA, and then like usually the mismatch repair genes will come along after your DNA is replicated and kind of go like, okay, there's a problem there. We'll go fix that problem before it proceeds, and we'll fix that problem before the cell cycle proceeds. But if you don't have that, then it will just continue into the rest of the cell cycle, because you can replicate your DNA, but it hasn't like your cell hasn't divided yet. So technically speaking, you could just hold the cell cycle and it remains one cell. It hasn't split yet. But if you don't have your mismatch repair genes, then it won't check your DNA, and then it will just continue and replicate. And with the mutations still there. But if you do have them, then it's all G. Does that make sense? I hope that makes sense. Oh, good. So yes, what is the two-hit hypothesis? It's not the most difficult question, but indeed, both alleles of the same gene must be affected. Not gonna lie, it took me a little while to actually understand what the two-hit hypothesis was, because they never really explained it very well in lectures. They were just like, yes, you need to have two mutations. I'm like, okay. But yes, both alleles of the same gene must be affected. So now we're going to talk about benign versus malignant tumors. So benign tumors, they look like the original tissue, but they tend to be like encased in this fibrous capsule. This stuff is actually decently high yield, because I mean, even in second year, we still talk about cancers and things like that. Like cancer will just keep coming back and coming back. Like they actually asked us a question about like does this tissue look cancerous and like malignant? Turned out it was actually just a kidney, but not the point. we were still trying to remember like all the features of cancer, whether they're benign or malignant. So yeah, they tend to look like the original tissue, and they don't spread. They're surrounded by a capsule, and they're generally not problematic at all, unless they're growing in a confined space. It's like if you had a brain cancer or like a benign tumor in the brain, even though it's not like actively invading, it's still just like taking up space and compressing the brain. And the brain's quite sensitive to like inflammation and things like that when it hits against the sides of your skull. Malignant tumors look angry and they don't look like the original tissue. So if you're seeing your normal tissue and there's just all these weird cells in it, it's probably malignant. And they have a high rate of metastasis. So when they're that malignant, they can basically just break off, go into the bloodstream, and spread throughout the body. And they are very problematic. And here are some pictures that I thought were kind of cool. So you can see your benign samples. Like you have your pink tissue, which is normal, and then you can clearly see like the defined edges of your kind of benign tumors, like all this. You can see like the clear definition between normal tissue and tumorous tissue. But here you kind of just have malignant cells just everywhere, because you can see the definition, but like it's so integrated that it's basically just kind of completely overtaken the normal tissue. So now we're going to talk about angiogenesis. So cancer, tumors can't become like invasive and cancerous unless it has enough blood supply. So because the cells are constantly like proliferating and they're undergoing like replication so frequently, that uses a lot of energy, and you need a lot of nutrients and like a lot of blood supply to actually support that. So yeah, so angiogenesis is actually something that's completely normal, but usually it's just like, you have some new tissue, or like as you're growing you get more tissue, and that tissue needs blood, and that's what usually happens. But angiogenesis, it can also increase blood supply to a tumor. So I think do I have a photo? Nope, don't worry. So like if you have your tumor and it's like in a part of the body that doesn't have much vasculature, it can like recruit, like it sends out, I can't remember what, I don't think it's particularly important to remember like what signals it sends out. I think it's a bit of like endothelial growth factor and stuff like that to promote blood vessels growing. But it basically just recruits like blood vessels to kind of come towards it and just supply it. So dormant cancer cells, so like if you had cancer had it removed and you're like, I think I'm fine. You could have like a small tumor that's just dormant, sitting in your tissue, and it doesn't grow or anything because like it doesn't have enough blood supply. But sometimes if it suddenly acquires a bunch of blood supply, like hormones change, or it suddenly manages to get enough blood, they can become active again. And they can, because they have more nutrients, and therefore they can proliferate like at a fast rate. Yeah. So also blood supply to a tumor is very crucial to its growth and like metastasis. So metastasis, I mean, I feel like you probably know this, but like it's basically spread of a tumor to other parts of the body. And this usually happens through the lymph nodes or the lymphatics or through the blood supply. Often veins, I feel. Like when you get to year 2, they talk about a lot of like metastasis to like the liver or the lung, because things tend to go back to those places quite a lot through the bloodstream. But yeah, angiogenesis. Important to remember. It comes up again in year 2, just like in passing. Like my ICL tutor asked me about angiogenesis the other week, and I was like, indeed, angiogenesis. So now we're going to talk a bit about metastasis. So it's invasion of cancer cells from a primary tumor to a secondary area. And there are 3 main stages of metastasis. So firstly, so you have like your cancer cell. I might just go here. So you have your cancer cell sitting here, right? So that's stage 1. This is like stage 0, kind of basically. The second stage is when they start releasing enzymes, and it like breaks through and it invades the basement membrane of the of whatever tissue it's in. So it breaks through the basal lamina. So because it doesn't have any of that anchorage dependence, it can break through. The next stage is they release some enzymes to like open up the capillaries more. So like the capillaries naturally have some pores in them, so like you can, give nutrients and stuff like that. But they kind of widen those pores so that they can enter the bloodstream and go along. So then they travel, blah 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 blah. They use bloodstream and lymph to travel. And then once they get to where they want to be, they will basically just stick to the side of the blood vessel and just slowly like squeeze their way through the through the capillary wall, and then take up residence in the new tissue. So that's basically it. Yeah, so then cancer cells, here we go. They secrete vascular endothelial growth factor. So that, if you just break it down, vascular, blood supply, endothelial, it's because blood vessels are all made of endothelial tissue. Like that is what makes up the walls of the blood vessel. And then growth factor, so it's promoting growth of blood vessel walls. And that promotes angiogenesis. And then, yes, they use it to grow the tumor, and then that is when it kind of colonizes that area. Okay, so now we've got a little case study. Breast cancer. So approximately 10% of breast cancer occurs due to genetic predisposition. That's a 10% again. And it's caused by BRCA1 mutations on the 17q arm of the chromosome, and the BRCA2 mutation on the 13q. So just as a refresher, 17 and 13 refers to like what number chromosome it is, so like 1 to, oh my god, 23. And then P, I think it's P and Q, and it's like the petite arm and the something arm. I'm not gonna lie, I can't really remember. After Sem 1, it kind of doesn't really matter because you tend to think more bigger picture after Sem 1 is over. I can't remember what Q stands for. But I think you remember it was like P is petite and the Q is like the other one, like the longer one. Yeah, so individuals with breast cancer are predisposed to ovarian cancer. So there's like a lot of relation between like cancer occurring in related systems. So like breast cancer and ovarian cancer are both like the sex organs. Why is breast cancer considered autosomal dominant? Oh god, my genetics knowledge. So I believe like if it's autosomal dominant, that kind of means like essentially, okay, I'm pretty sure autosomal dominant like essentially in essence means like you're going to get it from like your mother. I don't know if Honey is watching this. If Honey, oh, Honey is here. Honey, if you're paying any attention, please help me. Honey did genetics for the PSV. I need to refresh my genetics information. But I believe it basically just means like if it's autosomal dominant, you're basically just gonna inherit it from your mother's side. Is kind of like what it means. Don't mind me, sorry. And obviously, if you have things like breast cancer and ovarian cancer, it's gonna do that. I don't think it doesn't mean that you need BRCA1 and BRCA2. I think either one of these would predispose you to breast cancer. Does that make sense? I think when I say like autosomal dominant, it's more that like I think it's like you have an increased risk of getting it if you even have just like one mutated gene, if that makes sense. Like even if you have one mutated gene, you have do I have more slides on this? No. So like even if you have one mutated gene, you're already increased like and predisposed to breast cancer. I don't think it means that you are going to get breast cancer, but like yeah, 40 to 70% chance. So 40 to 70% penetrance rate basically means like if you have the gene mutated, then you have 40 to 70% chance of getting breast cancer. I think that's why a lot of people who like when they get their blood tested and they see that they have the particular gene, which is BRCA1 or BRCA2, they sometimes just like get the mastectomy and they just completely remove their breasts because they're like the risk is so high that like it's a very high risk of getting breast cancer, if that makes sense. Does that kind of answer your question? Cool. I mean, I'm happy to do some more research on it and kind of get back to you on it because it can be a bit confusing with the whole pedigrees and things like that. But to be honest, like with the whole pedigree thing, it's not really that important for like clinical practice because you do ask family history, but it's kind of more like, oh, his father, had lung cancer and his mother died of breast cancer. Then it's like, well, he's probably pretty likely to have cancer. So like you don't really think about it like in too much detail later on. Yeah, so other indications that you could inherit breast cancer or all these other things. So family history of breast ovarian cancer, obviously. Bilateral breast cancer, I think in like first and second degree family members. If you have ovarian cancer, you're very likely to have breast cancer. And early age onset, not gonna lie, I can't remember what that was. I think it's early age onset of puberty, maybe. Not gonna lie, I can't remember. I'm sorry. But I don't think it's that important. And also, if you have like anyone in your family that has male breast cancer, because male breast cancer is like really rare. So if they have it, then it's like, yeah. Oh wait, no, sorry, my brain's not really functioning. This is like if you have cancer with these factors, then it's indicated that it could be an inherited breast cancer. So like if you have bilateral breast cancer, if you have breast cancer with ovarian cancer, if you have cancer at quite a young age, then it's indicative that it was probably due to inherited breast cancer and not just natural like factors breast cancer. Sorry, it's been a little while since I made these slides. Okay, screening tests. So these are quite important. It's important to know that screening tests are always secondary prevention. They ask so many exam questions on this, and it's always just secondary prevention because they're always like, what kind of prevention is breast cancer screening or like prostate cancer screening or like all these various types of screening, and it's always secondary prevention. So just remember that. Yes, and so it's crucial to know that genetic predisposition to cancer, so that screening tests can be done more frequently. So like if you know that 3 of their family members have had bowel cancer, then you're going to want to screen them for bowel cancer pretty frequently. So yeah, so that can include gene testing, which is honestly like quite a lot of money and quite expensive, so not many people do that. Or looking for early evidence, so that's like colonic polyps, we'll talk more about colon polyps in a second, or breast lumps. Yeah, but you always must keep in mind like the emotional and physical effect on the patient, because you could take like, you could be like, oh, you might have cancer, and the patient might freak out a bit, and also like if you're taking a biopsy and the patient is like a little bit, immunosuppressed or something, that may not be the best idea because it's like a little bit invasive, or if they, don't clot very well and might bleed everywhere, then maybe don't take a biopsy, that kind of thing. So we're gonna look at this case study. I remember I remember sitting in the lecture theater listening to the lecturer go through this case study, and I was like, I don't understand what the fuck is going on. Like, I didn't think it was important. I didn't think like we would have to remember it, but I think it's actually a little bit like somewhat high yield. Like, they like to ask you about tyrosine kinase and stuff like that. So here we go. Here's a bit of an explanation about what everything is. It's honestly a lot simpler than the lecturer makes it out to be. So basically, tyrosine kinase receptors are receptors that sit in your membrane. So they are transmembrane receptors because they stick outside and they stick inside. And these transduce the signals of extracellular growth factors. So if you have your growth factor here, it'll come along, bind to the receptor, and then this receptor will send signals inside the cell. So it's outside to inside, transmembrane. And then these signals will, increase gene transcription, and then it should be like a negative feedback cycle where it then inhibits the intracellular signals. If you overexpress these, so if you have more receptors that are receiving more growth factors, then they are relaying more growth signals to the nucleus, then you will get increased production of cells. You will have blah, blah, yeah, so it can cause cellular transformation because it is increasing the rate of proliferation. So tyrosine kinase receptors are a type of oncogene because obviously oncogenes are genes that promote cell division. This obviously promotes cell division because it is transducing growth factor signals. Okay, so chronic myelogenous leukemia. It is a cancer, obviously, and very specifically, it is caused by translocation of chromosomes 9 and 22. Now, I feel like I remember like a practice exam question that actually asked you like what branches, like what two things like swapped. I don't know if it was for this or like for a different thing, like a Philadelphia something. But yeah, so basically what happens is that you have the 9 and your 22, and your 22 is already like a little nubbin there. And these two swap. So you have, okay, hold on. There are two genes. You have the genes ABL tyrosine kinase, and that sits on the 9. And you have BCR, which sits on the 22. Separately, they both play like roles in cell cycle regulation. So ABL plays a major role in cell cycle regulation. As soon as you put it next to BCR, ABL becomes overactive. So this swapping increases, so it's like a gain of function mutation of the ABL tyrosine kinase. So it becomes overactive and it can cause uncontrolled cell proliferation, which can become cancer. Yes, so this fusion protein basically contains a higher than usual amount of tyrosine kinase than normal ABL does. So ABL like somewhat helps in the like production of tyrosine kinase, and as we said, tyrosine kinase, if you have more tyrosine kinase, then you're going to have more growth. So if you have this gene, the fusion protein from these two combining, then you're going to have more tyrosine kinase, and that is what causes CML. And there are a bunch of anti-tyrosine drugs. I put them all here. I don't really think it's that important. I just put it in, for the sake of it. I guess it's more of a way of thinking about like how you can, like, what is the crucial parts of cancer. It's like ATP competitive inhibitors. Like, cancer needs energy to grow. So if you inhibit ATP receptors, then ta-da. Antibodies against receptor tyrosine kinases or their ligands. I mean, it basically just, antibodies against tyrosine kinase. And anti-angiogenics, I think that's a pretty decent point because like if you don't have angiogenesis, then you can't have cancer growth. So that's pretty good. You don't need to remember these. It's just kind of like extra knowledge to help stimulate your brain, I guess. Don't bother remembering it. Now we have another case study, and it's a retinoblastoma, and I think you do get quite a lot of questions about this because it relates to RB. And as you remember, RB is the gene that if it's mutated, I mean, you're kind of fucked. Like, I think it's mutated in like, what did I say about it? Like a very high percentage of cancers, basically. But it's named RB after retinoblastoma, because if you have a two-hit hypothesis of RB, then you can get retinoblastoma. So retinoblastoma is a rare childhood tumor of the retina in your eye, and it affects 1 out of 20,000 children. And you can have hereditary or non-hereditary retinoblastoma. If it is hereditary, then it develops at a young age. You generally get it in both eyes, and the mutant allele is on every cell of your body, which really sucks. But if you have non-hereditary retinoblastoma, it tends to develop at a later age and is usually only in one eye, and the chromosomal changes are only in the tumor cell. I feel like these parts are like the most important differences, because you have chromosomal change just within the tumor or chromosomal change in just your entire body. So this person would definitely be like higher risk of developing cancer because they have mutant RB alleles just everywhere. And lastly, we're going to talk about, lastly, we're going to talk about colon cancer. So this actually does come back in year 2. We were talking about it the other week because we had an ICL case about a guy that has bowel cancer. So we're going to talk about familial adenomatous polyposis. And that's just like absolutely disgusting 3 words. But basically it's polyposis is just having a shit ton of polyps, like all over your small intestine. So this is what it normally looks like. This is what it looks like when you have polyposis. So usually when you have like polyps in your normal intestine, you would have like 1 or 2 polyps, and then they're like, oh, that could be cancer, and then they'll take it out. It becomes a lot harder when your entire intestine is just polyps, and you don't know which polyps are benign and which polyps are cancerous, and it just basically, since you have so many polyps, there's a higher likelihood that at least one of those polyps are going to be cancerous. Yeah, so polyposis for FAP, yes, is caused by a mutation in a tumor suppressor gene called APC. So it's a tumor suppressor gene mutation, so it's a loss of function mutation. So APC is responsible for cell adhesion, signal transduction, and activation of RAS. I'm not gonna lie, I don't know what that is. I don't think it's that important to remember. None of these case studies are like super important to remember every detail of it. It's generally just like how to put it into like to help you visualize what the theory is in real life. But yeah, it's almost 100% penetrance, because if you have this many polyps, it is very likely that at least one of them will be cancerous. And you can't go along and remove all of the polyps because they are just everywhere. It would take hours and hours, and then they would reappear again, so it's completely useless. Yeah, so if they become malignant, they must be removed. And because you don't have like the normal structure of cells, like you don't have your villi and all that, it can cause malabsorption of nutrients. So now we're going to talk about the effects of cancer on a patient. I'm sorry this is like such a long presentation, but we're getting there. Okay, so some local effects is that firstly, it will displace some normal adjacent tissue. That makes sense. You have loss of function. So basically your normal cells that have, their specialized functions. So say you had like your cilia in your throat generally like move mucus up so you can cough it up. If you had like a esophageal cancer, those ciliated cells won't have cilia. So they can't perform their normal function. They can also block lumen of passages. So like if you have a colon cancer that grows and blocks the bowel, it can, affect your GIT movements and like your blood supply and stuff like that. And that can cause like infection and all these bad things, because if you have like poo stuck in your small bowel and then you have all the bacteria in your bowel and they just like congregate there, then it can cause infection. Same thing with blood vessel or lymph, and that obviously is very bad. So then cancer cells also compete with normal cells for nutrition and blood supply, and it can mean that other organs break down their own proteins to nourish the cancer, so patient may lose a lot of weight. So that's why they say usually like if a person has come in to the clinic and they say like, oh, I've lost like 10 kilograms in the past like 5 months, that's quite concerning, because if you ever have a patient that comes in with like an insane amount of weight loss, you always think cancer. Like cancer is always on your differentials, because of this thing where cancer cells will like basically steal nutrients from other cells and your other cells will break down in order to try and nourish the cancer. So inheritance, look, I feel like we've talked about inheritance to death already. But yeah, but an important thing I think that's quite interesting to think about is that you can have familial aggregation partially because of genetic predisposition, but also because, like, if you grow up in the same household as your mother and the house has asbestos, then you're both likely to get cancer. Um, Penetrance is the percentage of individuals with the affected gene who develop the cancer. So we've kind of discussed this already. So, like, colon cancer can be 100%, breast cancer can be 40 to 70%. And if you have first degree relatives, or like a lot of close family members of that, like, that have cancer, then it's probably going to be inherited. And you need to know this for cancer screening and family counseling. We've kind of gone through that already. And I'm done. And now we are going to pass over to Jess to talk about blood components.
Ooh, okay, cool. Honestly, I'm happy for you to keep sharing. You can just click through it. I don't have that many slides.
Okay, cool.
Okay, so hematopoiesis. So this basically is the formation of any blood cellular components, which includes like your platelets, red blood cells, white blood cells. So this entire process occurs outside of the bone marrow, which is also called extramedullary before you're born, and the production of this stuff ends up in the medullary cavity, which is the bone marrow cavity in your long bones. So like between like the spongy bone. I think you guys will go through like bone structure a bit more later. But that's where it is after you're born, and that's where most of our blood cells are produced now. So these two things probably aren't super important, but I think that one thing that you should really take away from hematopoiesis is that iron, folic acid, and vitamin B12 are three things that are really important for the production of cells. And I think, I'm not 100% sure, but I think this is in reference to like red blood cells in particular. So I had a friend that told me that the way he remembered this was by thinking about IFB as like I fuck bitches. I don't really, it's a bit much, but if it helps you, it helps. Okay, so like the first thing I'm gonna go through on this slide is like this blood histology kind of thing with the staining. So it's kind of worth just knowing what each thing looks like on the slide. So you've got the red blood cells with a very classic biconcave appearance with like a kind of more see-through inside and like a ring around the outside. And it's pretty important to be able to like identify what these look like on histology slides just because they come up again and again. Um, and then you've also got the white blood cells. So you can see that you've got like a lymphocyte with a really big nucleus. You've got a monocyte, which is slightly bigger. They look fairly similar though. And then you've got a basophil, which is fairly identifiable because it's got all these granules in the middle and just everywhere. Just you can see little dots, and that's because the basophil destroys things by releasing like toxic granules. And yeah, you can really see them here. And you've also got eosinophil and neutrophils with like multiple nuclei looking bits in them, and that's how you identify and separate them from like the lymphocytes and the basophils. You can also see a little platelet here. It's small boy, and it's usually like the smallest thing in like a slide if you see that. Okay, and in terms of like the diagram on the right, you've got hematopoietic cells, which can lead to differentiate into two different things. So either a common myeloid progenitor or a common lymphoid progenitor. And the myeloid ones usually lead to things that are more in like the innate immune system, so like eosinophils, neutrophils, basophils, monocytes. Whereas the lymphoid progenitors lead more into like the adaptive immune cells. And you can go back to our immune system slides slash presentation just to have a look at like what exactly originates from which progenitor, because this isn't the full list. But what is important for here is that you see that platelets and red blood cells both come from the myeloid progenitor, and they come from like megakaryoblast or proerythroblast, which is just like the step before they become full like matured cells to be circulated in the body. Okay, so the next slide is this little diagram that shows you what's actually inside the blood. Okay, so you've got, hang on, sorry, someone just asked about if histology is important. I would say it's probably so it comes up again and again, and they really try to hammer it home like what you can see on a histology slide and like what things usually look like, and it's not they don't ask a lot of questions about it, but because they keep talking about it again and again, you end up just knowing it anyways. And I think that Like it's a lot to take in at first, but because you do it, in first sem, for second sem, you do it again in second year, eventually you just kind of know to like recognize things, and it's a really big learning curve, but you'll get there eventually. Okay, yeah. So back onto the components of the blood. So you just need to know that there are three parts. Like the percentages that I've listed here probably aren't that important. They just kind of help you picture what is in the blood. So you've got the plasma with all of the things minus the white blood cell, platelets, and red blood cells. So these three important cells, which I usually associate with like blood and like important bits of the blood, but they're not in the plasma. And like sure you can go remember what exactly is in each of these parts, but I also don't think that's super important. Okay, erythrocytes. So these are your red blood cells. So from the development of like that undifferentiated cell, it takes 3 to 5 days for the red blood cells to actually form. The function of this is to transport oxygen, so it's like specialized in a way to be able to do that. So for as an example, it doesn't have a nucleus and doesn't have any organelles. These are both removed before the cell is released into the bloodstream. So it's worth knowing that they do have a nucleus and organelles as they are developing, but these are removed as they mature. So because they don't have this nucleus, they're not able to undergo like regular metabolic processes. And the only way they can get energy, like ATP, is by metabolizing glucose by glycolysis, which doesn't require oxygen. Sorry, not oxygen. Doesn't require a nucleus or like, any organelles. And this is again for the purpose of being able to transport gases. There are a couple like numbers listed here, like 3 to 5 days, 120 days in circulation, 78 micrometers. Like it might be worth like knowing approximately what these numbers are, but I think there was like a quiz question on it, but I don't think it necessarily came up in the exam. And another thing that you should probably know is that erythropoietin stimulates differentiation of stem cells into red blood cells. So this is just like an important thing that helps this process of maturation happen. And depending on the levels of red blood cells in your body, you can get more of this to stimulate more red blood cells, and that maintains the level in your body. Okay, so erythrocytes and hemoglobin. So we've got two kind of parts to this slide. So the hemoglobin itself is 4 globin chains and a heme group containing iron. So this kind of comes together. This is like a big mess, like that spaghetti looking thing in the previous slide. That is, that's basically what makes up the red blood cell. And so there are a couple factors that affect how tightly the oxygen binds to the hemoglobin. So an example of this is like your 2,3-DPG. There's a little bit of a typo there. It's DPG. And you definitely come back to this again, but I don't know why, but this stuck with me in first year. And it's just basically a thing that determines how tightly your oxygen binds to your hemoglobin. So if you have more of it, it binds less tightly. So you can change like your oxygen uptake with the hemoglobin just by changing this 2,3-DPG. And this second part of this slide is this diagram here. And it basically just shows how hemoglobin is broken down in the body. So this diagram is definitely super confusing, and it's probably not really worth like really trying to understand now, because you come back to it like in second year, and it makes a lot more sense now that we've covered like all the stuff that's relevant to like understanding how hemoglobin's broken down. But I think the gist of it is that you've got a heme part and a globin part, and the globin part is recycled in the plasma protein pool, and the heme part is further broken down into like biliverdin, CO, and Fe, so carbon monoxide and iron. And the iron is again recycled again. It goes back into the bone marrow, and it undergoes multiple processes. I think what's less important here is that, like, you'll learn it later, but it's just this biliverdin, bilirubin stuff, because it just gets excreted or like does some other funky stuff. But I think the main idea that you're trying to take away from this is that there are certain parts of the hemoglobin that are recycled, so you don't have to be making them again and again. Once they're detached, they just reattached onto like a new molecule, and it's fine to go back into the system for circulation. Okay, megakaryocytes. So these are like really big cells that the cytoplasm is what forms the platelets. So they have like the bits of cytoplasm just come off, and they become platelets. And these are regulated by thrombopoietin. Another like keyword maybe to remember that platelets, thrombopoietin. There isn't really much else to this, I don't think. They're produced in the bone marrow as well, like a lot of the other cells in the body. Okay, so abnormalities. There's a lot of new words here, and I definitely feel like it's easy to feel overwhelmed, especially when you're seeing these for the first time. But there's a couple ways. So I think the main thing is just to remember the prefixes and or suffixes of these words that will tell you about like what it actually means. So anemia is like reduction of hemoglobin. I think emia supposed to refer to blood cells. Um, and poly is like too much. So this kind of just relates to like the meaning. So reduction of hemoglobin, increase in hemoglobin. Um, and then leukopenia, I think penia is low levels, and cytosis plus philia mean like high levels. And once you like put these words together, you can figure out what it's referring to. And like you haven't learned about a lot of the clinical like relevance of these things yet, but knowing what they mean helps you understand what's happening with the patient and what's going on with the blood in their body. Yeah, so I don't think it's too much of a like you probably shouldn't go through this and try to remember all of it, but it's worth just knowing these like little bits of information where you can figure out if it's, oh yeah, this person's got high levels of white blood cells, if this person has got high levels of like neutrophils or low levels of monocytes, and that you can then link that into important things in the future. Okay, so full blood counts. So these are taught in pre-clin. I don't think anyone really remembers it because it's like that we it's hard, it hasn't been associated with anything that's particularly relevant. So, and I really don't feel like it's that examinable just because like in pre-clin, like what are they gonna do? Like this, it's just there. Um, but it is very much like very relevant in your clinical years. Like once you hit third year, I think you probably should know like what each of these are and like what is in the full blood count. Definitely for like first and second year, like I don't I personally wouldn't recommend like going and memorizing exactly what's here. Can if you want, but I just don't feel like it's that worth your time just because it's like hard to link back into anything that's like important. But the counts themselves are this is the full list, and the brackets kind of just explain what the the actual test is. Um, and some of these are fairly self-explanatory as well. Like the mean cell hemoglobin is just how much hemoglobin is in each cell on average. So yeah, yeah, that's pretty much it then for blood components. This is definitely a tough one because there's a lot of content, and a lot of it definitely is a bit. It's because there's no context to it. It's hard to remember, and it doesn't feel like. I just don't feel like it's that example in first year. Like it's very important. It definitely is very important, but I think this is stuff that you revisit again in the future, and it becomes more and more relevant as you like associate with something clinical or something. Yeah. Okay. Anyways, that's pretty much it. Thanks for coming.